Hello, this is Bronwyn Ailey, Local Food Systems and Small Farms Educator for the University of Illinois Extension. And for this week's update on local food happenings at DSAC, I wanted to focus on soil testing that we're going to be doing in one of our high tunnels. As you know, we have, as I have mentioned in the past, we have 3 commercial-size high tunnels here, and one of them is in hydroponic production. One of them is, and the other 2 have in-ground raised beds, permanent raised beds. And the other tunnel is currently in production with tomatoes and peppers, cut flowers. And this 3rd tunnel we have been utilizing, we utilized last year for a Illinois Specialty Crop Block Grant that we received to look at winter vegetable production. And so we have left this tunnel empty during the summer season so that we can get it prepped and be ready to start planting in late August and September for the 2nd year of that grant. And so one of the things that I wanted, a task I need to accomplish before we plant this tunnel, is I need to go through and gather soil samples from each of the rows so that I can get an idea of the fertility levels through each row, so that I know what I may need to apply prior to planting the fall crops. And so I'm going to just go through some real basic steps on taking a soil sample. Most people have seen other videos on soil sampling, but I just wanted to show you one of the aspects that we're going to go through in preparation for the 2nd year of this research grant. So the basic tools or equipment that I'm going to use for the soil sampling is soil probe. I prefer to buy the ones that have the little step right here, so that when you're in taking a soil sample in soils that are either really wet or dense or really compacted, being able to step down on this and put pressure on it really helps me to gather a better sample. Also, just have a basic 1-gallon bucket here that I'm going to put the core samples in and mix together before I put into I just had Ziploc bags on hand, so that's what I'm going to use. You can use brown paper lunch bags, like I said, Ziploc bags, even plastic Walmart bags would work. Just something that you can collect the sample in before you send it off to the lab. And so another thing to remember is when you're submitting multiple samples into a lab, you need to make sure and label or mark the samples in a fashion that you will know when you get your test results back where those samples were taken from. And as an example, in this tunnel we have 8 beds across the tunnel, and I'm actually going to take cores, 4 core samples from each bed, and I will label them. This on my labeling I have S1 because this tunnel, the high tunnel itself, was manufactured by a company called Stuppy, and so we have always referred to this tunnel as the Stuppy tunnel, so I'm calling it S1. So row 1, which would be as I walk in the door and look to my left, I'm going to consider that row 1, and I'll go S1 through S8, and each row will have its own bag. And we'll go through and collect core samples, 4 cores down each row, put them in this bucket, mix them together, and then put maybe half a bag's worth. This is a 1-quart bag. I'll put maybe half a bag worth of soil in here to send off. The soil testing labs they need a sufficient amount, but not an overabundance of soil. It also makes, if you're sending in several samples, it can make your shipping kind of pricey. The heavier the samples are, and the more you include in that. And so we don't need to be sending a ton of extra soil to the lab that they don't need, but we do want to make sure we're providing them a sufficient amount. So about a pint per sample should be ample sample material for the lab to run the tests. I guess one other comment I would like to make about the when you're submitting a sample into your lab, you want to make sure and let them know what crop you are planning to plant into the area that you're sampling. A lot of times if we don't mark that, the soil testing labs will send us back recommendations that could be based on more row crop crops instead of specialty crops. If you think about the state of Illinois in general, we are definitely known for our corn and bean production, and soil fertilizer recommendations for corn and soybeans are a lot different than, say, tomatoes or even the winter greens that we'll be growing in this tunnel later this season. And so you need to keep that in mind and maybe make a note of that when you're sending that sample in, so that the lab knows what crop you are planning to plant into an area. And some labs do a really good job of making recommendations for different specialty crops as well. So with that, I will get started collecting a few samples down this row. So I'm going to take 4 samples in a row, just kind of divide it, estimate about a quarter of each row. I'm going to put my probe, not on this very edge, but somewhere in the middle third of the bed. Push it in, give it a twist. I'm going to go ahead and step on it, make sure it gets all the way down in there. I twist it out, twist it up, and I'll come in closer so you can see. We have a full 8 to 10 inches of soil collected here. So I'm going to dump this into my bucket. The probe I can push on the bottom and push the sample up out of the tube into the bucket. It's amazing, this tunnel is about 110 degrees right now, and the top couple of inches of the soil was very dry looking, but as you can see, there's plenty of moisture still underneath. The core stayed together. So I'm going to show you that. So that was the first sample. I'll go ahead and take 2 or 3 more here. Okay, now that I've collected 4 core samples from row 3 here, I'm going to, and again, we'll look at the bucket. You can see moisture in all of the cores, if you can see that. I'm going to mix this up, kind of break the moist cores up. I want to make sure I'm sending in a good representative sample from each area that I sample, and that's why we take multiple cores per sample size. If I was to just take one core sample from each row, this may not be a good representation of the whole row itself. And so even just a 90-foot row like this, I want to take multiple samples, cores, and mix those together. So I've kind of broken them apart and mixed this up, and I'm in row 3, so I'm going to grab my sample bag that's already labeled with S3. Open that up, and we'll just pour soil in here. Make sure you don't drop it. Have to recalculate. And it looks like about 4 cores gives me right at that half a bag level. So that works out really well. Kind of squeeze that tight. Make sure it's sealed good. And this sample will be ready for me to send off. And so I'll finish collecting samples from each of the rows. And once I get all those collected and put together, I'll be sending them in either today or the first of next week off to the lab, so I can get my results back in time to apply fertilizer as needed on these rows in preparation for our winter vegetable production in this tunnel. As it's been 2 or 3 weeks since I've given you an update on the progress of our different crops growing here in the tunnels at Dixon Springs, I thought I'd just do a quick walkthrough and show you the progress of our tomatoes and peppers. We began harvesting, actually I guess the first of this week, and you can see we actually have harvested today already as well. You see some fruit that are definitely ripening. We like to have the fruit hang on the plants for just a little bit longer. That doesn't necessarily mean that should be the course that everyone would take, but for our purposes here, we like to let the fruit hang just a little bit longer and ripen a little bit more completely, not 100% on the vine, but pretty completely. And you can see a lot of the tomatoes are now growing taller than the stakes. We can no longer see the names on them anymore, starting to grow together. We've got room to probably put one more string on to try to squeeze them back together in their row. But they're definitely coming on. The fruit size is tremendous this year on these tomatoes. And then on the bell peppers, we've probably been picking them a couple of weeks now. Again, this is the spacing study. Same variety, just laid out in different spacing. And then this row is a mix of just different types of peppers. This Sweetie blend is like the little snack peppers. We also have a banana, poblano, and a jalapeno. And then the final row over here would be our cut flowers. And we've been going through about once a week and cutting back flowers. Just depends. We really cut back the larkspur, and it's starting to come back. Not nearly as tall as it was before. The zinnias are coming on really well. They're in the very back. The snapdragons have come back. So we're plugging along here, generally harvesting and grading something every day. This tunnel, we've been doing pretty well on our insect management here. We continue to scout every day. We may see a few thrips in this tunnel, but nothing major. I haven't seen very many aphids lately, so we're keeping those in check. And we've maybe found 2 or 3 worms, a couple of tomato worms and a couple of army worms, but really nothing of significance at this point for this tunnel. And we'll also just do a quick update on the crops being grown here in the hydroponic tunnel. You can see the cucumbers. We're working on, they're a continuous process of pruning, harvesting, lowering. You can see the vines are starting to wind around on the ground at the bottom where we've pruned off the leaves and suckers, and want to have about 4 to 5 feet of foliage left on the vine, and we can just continue to lower it and wind that stem down at the ground. And then also you see our bell peppers being strung up. We've gone through and harvested these earlier this week. And also our indeterminate indeterminate tomatoes. We're using the same system that we are with the cucumbers as far as the lowering. These are growing at a slower rate, and so you don't see the stems winding down as much on these particular varieties. But then when we look at these the other set of varieties on these tomatoes that are more greenhouse style tomatoes, you can see that we have a lot more vine growth on these. You can see the stems starting to lay on the ground, and We've gone through and pruned and lowered those earlier in the week. It looks like we need to get in here and harvest today yet in this tunnel. I will note that the thrips are really high. We have a high population of thrips in this tunnel, and so one of the cultural practices that we will engage in is that we do not come into this tunnel or work with these plants until we're done in the other tunnel, so that we don't move insects into the other tunnel that aren't already there. The thrips pressure is extremely high in here, and so we don't want to transfer any more of those out into the other tunnel. So work in this tunnel is done after we've completed other tasks in the other tunnels, so that we're not transferring any potential insects over there. But that gives you an update on where we're at in the hydroponic tunnel as well. We're experiencing our typical July Southern Illinois summer, so we have 90 plus days with extremely high humidity, and so our heat indexes are running at above 100. So I want to remind everybody, when you're working outside, to keep your safety in mind, stay hydrated, take more breaks, cool off, get in the shade. Just watch out, because this heat can really zap you if you're not careful. And with that, we will be bringing you another update next week.